Hello everyone, welcome back. Today I am here to discuss about this important topic of cell biology, that is nucleus, commonly known as director of the cell. In Latin word nux, which means nucleus or center or heart of the cell. It contains DNA, and in Greek we use a synonymous term that is called karyon for this nucleus. So these are the terms which are used for this nucleus, and it controls the activity of the cell. That's why it is termed as the director of the cell. In this presentation, I will discuss about the historical background of nucleus, its occurrence, position of nucleus, its number and shape, size of nucleus, its chemical composition, and the ultrastructure of nucleus, which is made up of nuclear membranes, then nuclear pore complex, nucleoplasm, and it contains one dark stain structure inside the nucleus, which is known as nucleolus. So these are the topics that I will try to cover in this presentation. Let's dive in. Firstly, historical background. Nucleus was first discovered and named by Robert Brown in 1833. This is the portrait of Robert Brown. Then this is the animated diagram of nucleus. It consists of various parts like nuclear envelope, chromatin. Nuclear envelope contains outer membrane and inner membrane. It also contains an internal matrix that is known as nucleoplasm, then nucleolus. This person is M.J. Schlinden, who first described the nucleoli. And this cell organelle, that is little nucleolus, that was first noted by Fontana in 1781. So nucleolus was first noted by Fontana in the eel skin cell. जो ईल का स्किन सेल है उसमें सबसे पहले नोट किया था फोंटेना ने न्यूक्लियोलस को. दिस इज फेलिस फोंटेना in 1781. It's 81. He investigated human eye. He described the nucleolus after he found it in an eel slime or eel skin cell. And the term nucleolus was coined by Bowman in 1840. जो Bowman है उन्होंने ये term coin किया nucleolus. Then Strasburger in 1882 introduced the term cytoplasm and nucleoplasm. So this is the photograph of Strasburger. And then the most important discovery, that is the role of nucleus in heredity, that was first established by a grafting experiment of Hammerling in 1953. This is the photograph of Hammerling. He first described that the nucleus plays an important role in heredity. He worked on one green algae, which is the largest plant cell, that is Acetabularia, umbrella shaped algae. So he used two species of Acetabularia. One is Acetabularia mediterranea, second was Acetabularia crenulata, and he removed their heads. जो इसके heads हैं उन्हें remove किया गया था. Then he swapped the stalks, और ये जो stalks हैं इनको exchange किया. So that we can see he found intermediate heads. जो heads उसमें बाद में बने after exchanging their stalks, these heads were intermediate, and there was a confusion that which substance controls the development of head. कौन सी ऐसी चीज़ है जो head की development को control करती है? It may be nucleus or some other chemical which is present in the cytoplasm. Then after that he further extended this experiment. He found that if intermediate heads are removed, अगर इन intermediate plant से heads को remove कर दिया जाए, so जो new heads grow करते हैं, that correspond to the nucleus of the rhizoids. तो वो जो heads होते हैं वो डिपेंड करते हैं जो rhizoid में nucleus है उसकी तरह head इसमें grow करेंगे. So we can see here this is the rhizoid of Acetabularia mediterranea, और इसमें जो cap है वो भी Acetabularia mediterranea की है. While in other case there is the rhizoid of Acetabularia crenulata, और इसमें जो cap है वो भी Acetabularia crenulata की है, irrespective of their stalks. So this demonstrates the role of nucleus and chemical messenger in the development of the cell. जो cell cap है उसकी development में nucleus का important role यहाँ पे explain किया गया था by Hammerling. The next scientist was Barbara McClintock. She reorganized and named nucleolar organizers. So जो NOR region है उनको उन्होंने recognize किया था. And the credit of discovery of jumping gene, that is transposon, that also goes to this scientist named as Barbara. If we talk about its occurrence and position, so nucleus is found in all the eukaryotic cell. Eu means true, karyons means nucleus. The cells having true nucleus are known as eukaryotes. But there are some exceptions, like we have mature RBCs and seed tube cells in case of plants. So these cells lacks this nucleus. So this cell organelle, which is the largest cell organelle, is found in all eukaryotic cells. सभी के सभी जो यूकैरियोटिक सेल्स हैं उनमें पाई जाती है of the plants as well as animals, and this is the director of cell. It directs the or we can say controls the activity of the cells. We have some exceptions, like we have mature seed tubes of higher plants and mature RBCs of mammals, जिसमें कि ये सेल ऑर्गेनली एब्सेंट होती है. RBCs में न्यूक्लियस इसलिए एब्सेंट होता है so that it can provide maximum space to carry oxygen. While in case of seed tube elements, जो कि फ्लोएम का पार्ट है, इनमें भी जो न्यूक्लियस है it is absent in seed tube, while it is present in the adjoining cell, which is also known as the sister cell, जिन्हें कहा जाता है companion cells, which control the activity of seed tube cell, while RBCs, which are enucleated. If we talk about its lifespan or shape, it is biconcave shape in case of mammals, and it's the most abundant cells in the body. Its amount is 5 to 5.5 millions RBCs per ml of the blood in healthy human, and its lifespan is of 120 days. The site, or we can say the graveyard of RBCs, is spleen. Spleen में ये RBCs destroy होती हैं. So that's why this spleen is also known as the graveyard of RBCs. And the corpuscles have no organelles, and mammalian RBCs are enucleated. It means they lack nucleus. So incipient nucleus, that is called the nucleoid or prokaryon or genophore, that is present in case of the primitive cells, which are known as the prokaryotic cell, as in case of bacteria. So ये जो बैक्टीरिया है, इसमें जो ये सेंटर है, that is occupied by this chromatin material or DNA which lies freely in the cytoplasm, और इसके लिए टर्म यूज़ किया था न्यूक्लियोइड, या फिर इसे कहा जाता है प्रोकैरियन, या फिर इसे कहा जाता है जीनोफोर, because this genetic material doesn't lie in the well membrane bonded nucleus. ये इसमें cytoplasm में lie करता है. So that's why these types of cells are known as prokaryotic cells. In addition to this, we have some other organisms, like we have green algae, blue-green algae, BGA. It is again a prokaryotic cell. Next comes its position. So its position is not fixed in all the cells. So we have different cells where its position varies. Generally, it is centric in position in embryonic cells. जो एम्ब्रियो के सेल्स हैं और एनिमल्स के सेल्स हैं, उसमें इसकी पोजीशन होती है सेंट्रल, मीन सेंटर में लाई करता है, while it is peripheral in adipocytes. Adipocytes means the fat storing cells. Then it is basal in columnar cells and the gland cells, जिसमें इसकी पोजीशन होती है basal. And it's eccentric in plants. जो plants हैं उनमें होती है इसकी पोजीशन. Eccentric means away from center, that we can see in this diagram. Why it is eccentric? It is because of the presence of this large size structure which is called vacuole. So जो vacuole है इसको push करता है away from the center. The next comes this number. So mostly जो cells होते हैं, they are monokaryotic, means it contains, every cell contains a single nucleus. But we have exceptions, like we have anucleated cells as in case of RBCs of mammals. Then we have binucleated cells, like we have civated protozoan like paramecium. Paramecium में दो nuclei होते हैं, one is macronucleus and other is a micronucleus. And we have some cells which are multinucleated, like in case of Populina and striated muscle fibers. इनमें एक से ज़्यादा nuclei present होते हैं. Then multinucleated cells are also termed as cenocytes or cenocytic cells, as in case of fungal hyphae of Mucor and Rhizopus. These are also present in case of algae, like we have Vaucheria. जो Vaucheria है उसमें present होते हैं. This is a algae, that is a xanthophoric algae. Xanthophoric means yellow-green algae. So this algae is again siphonaceous, or cenocytic means a cell having multiple nuclei without any septa. In animal cells, we use the term for this type of cell as syncytial, for example, epidermis of nematodes. Then we have prokaryotic cells of polykaryotic cells of mussels, contains hundreds of nuclei. Next comes its size. So its size is again not universal in every cell. It varies from cells to cell, organism to organism, and activity that are carried out by the nucleus. So nucleus cytoplasmic index, that is called NP ratio, that lies between the size of nucleus and amount of cytoplasm. So दो factors हैं जो इसे determine करते हैं, that is size of nucleus and amount of cytoplasm. And mathematically it is represented by NP is equal to Vn divided by Vc minus Vn, where Vn is equal to volume of nucleus, Vc is equal to volume of cytoplasm, and nuclear volume increases with increase in cytoplasmic volume. The next comes the number of chromosome. As the size of nucleus is smaller in case of haploid cell, while it is larger in case of diploid cell, and it is again greater in size in polykaryotic cells or polyploid cells. So nucleus of polyploidy is greater in volume than that of a diploid cell, which in turn is larger than the haploid cell. Next comes this metabolic activity. So metabolic activity also define the size of nucleus. So larger in size in active cells. So nucleus का जो size है वो बड़ा होता है in active cells than as in case of resting cell as compared to the resting cells. Resting cells में nucleus का size होता है छोटा. Next comes shapes of nucleus. So its shape again, mostly in cells, its shape is spherical or ellipsoid, but its shape varies in some cells. So generally spheroid, but ellipsoid or flattened nuclei occurs in some cells. We have some exceptions, like we have different types of blood cells, WBCs, that is white blood cells. In case of neutrophils, its shape is multi-lobed. This is multi-lobed, not multinucleated. So we can see in neutrophils, the shape of nucleus is multi-lobed, while in case of acidophils or eosinophils, that is its shape is bilobed, while in case of basophils, again it may be bi to trilobed. In case of monocytes, its shape is kidney-shaped. So it contains kidney-shaped nucleus, while in case of lymphocytes, it is circular in shape and it is larger in size. While in thrombocytes, it is eccentric. So these are the various types of nucleus shape present in different blood cells. Okay, next comes this chemical composition. So chemically it is made up of proteins, DNA, RNA, and lipids. So it contains 80% protein, 12% DNA, 5% RNA, and 3% lipids. And it contains some structural proteins, basic proteins like we have histones, or as constituents of several enzymes. Constituents of several enzymes means the proteins which helps in formation of many enzymes. So these are also found in the nucleus. So traces of minerals, which are very important for replication or other processes which are carried out inside the nucleus, like we have calcium, sodium, potassium, magnesium, phosphorus, etc. Next comes this ultrastructure. So nucleus is formed of 4 components, like we have firstly nuclear membranes, termed as karyotheca or nuclear envelope से कहते हैं. So this is a double membranous system. 2 membranes होती हैं इसमें. That is outer membrane or inner membrane. That is outer membrane is known as ectokaryotheca, and the inner membrane is known as endokaryotheca. So these are the terms used for this nuclear envelopment, and it forms a boundary between cytoplasm and nucleus. The next important structure is nucleolus. Next we have this nuclear sap or nuclear matrix, जिसमें present होता है DNA. Then next we have this important structure that is called nuclear chromatin, which is basically DNA that controls the activities of the cell. So firstly, nuclear membrane or karyotheca. So outermost thin semi-permeable bilayer envelope of interphase nucleus, it is present in the interphase nucleus, while when the cell undergoes cell division, this nuclear envelope's destroyed or disintegrates, so that there is separation of the chromosomes. And its main function is in regulation of nucleocytoplasmic interactions. It separates nucleoplasm from cytoplasm, so it acts as a boundary between nucleus and cytoplasm, and it is commonly known as nucleolema or karyotheca. ये terms इसके लिए यूज़ की जाती हैं. This was discovered by Erklab in 1845. इन्होंने सबसे पहले इसे डिस्कवर किया था. And it is present in almost all the eukaryotes. जो eukaryotic cells हैं उन सब में ये membrane present होती है, and it is absent in prokaryotes. Next comes structure of this membrane. So nuclear envelope is a bilayered envelope formed of two membranes lie more or less parallel to each other. दो membranes present होती है इसमें, and each one is about 75 Armstrong thick. तो जो membrane की thickness है वो है लगभग 75 Armstrong and is lipoproteinaceous and trilaminar as so it shows similarity with the plasma membrane. And this outer layer is known as ectokaryotheca, while the inner one is known as endokaryotheca, and it lies in contact with cytoplasm. So this outer layer, that is outer membrane, it lies in contact with cytoplasm, and it is studded with ribosomes because it forms a continuous connection with the endoplasmic reticulum. It means it's a part of RER, that is rough endoplasmic reticulum at some points. Next is perinuclear space. So this is the space which lies between these two membranes, that is ectokaryotheca and endokaryotheca. So दो जो membranes हैं इनके बीच का जो space है, this space is known as perinuclear space. This is a fluid filled space lying between two nuclear membranes, and it is about 100 to 300 Armstrong thick. इसकी जो thickness है इस space की वो है लगभग 100 to 300 Armstrong thick, and it may have fibers, antibodies and crystalline bodies. तो ये इसमें present होता है, and it is a continuous with ER and plasma reticulum साथ continuation में है, and lumen and is engaged in protein synthesis. Next comes inner membrane, that is commonly known as endokaryotheca. So inner membrane is termed as endokaryotheca, and it lacks ribosome featuring an electron dense fibrous lamina or nuclear cortex. और इसके साथ जो fibrous structure present होता है उसे बोलते हैं nuclear lamina. This was discovered by Harris and James, and it forms a honeycomb layer within the nucleoplasm. ये बनाता है एक layer within the nucleoplasm, and it is comprising of special proteins जिन्हें कहा जाता है laminin proteins. Laminin proteins का जो ratio है, जो laminin proteins है इसमें दो तरह की laminin proteins present होती है, A and B. जो A lamins हैं वो बनी होती है A और C से, and while this B type of laminin protein is made up of B1 and B2 and are associated with membrane proteins. It maintains nuclear structure and integrity. जो lamins हैं वो important role play करती है to maintain the integrity of nuclear membranes. Next, the inner nuclear membrane houses lamin B. जो inner nuclear membrane होती है इसमें होता है lamin B के receptors and facilitating interaction with lamin A and C, जो कि facilitate करते हैं interaction of lamin A and C. So lamin proteins are formed by type 5 intermediate filaments. There are some filaments which are known as type 5 intermediate filaments. ये बनाते हैं lamin proteins को and compose the nuclear lamina, जिससे बनता है nuclear lamina, regulated by phosphorylation and dephosphorylation reactions. तो इसकी formation में दो reactions important role play करती है, that is phosphorylation and dephosphorylation reactions. Lamins का जो main work है, that is to support nuclear structure, its shape and chromatin attachment. So these are the functions carried out by these proteins which are known as lamins. So phosphorylation triggers nuclear lamina disassembly. जब phosphorylation होती है तो nuclear lamina की disassembly होती है, aiding in nuclear envelope breakdown, जो कि cell division के time बहुत important role play करता है for disassembly of nuclear membrane and reformation during cell division. और जो reformation होती है cell division में वो भी होती है इन्हीं reaction से. Next important structure which lies in nuclear membrane is nuclear pore complex. As the name indicates, pore complex, it means this pore is made up of different proteins, and its main role is in controlling the flow of material inside or outside the nucleus. So the nuclear envelope featuring small perforations allowing free passage of small molecules and ions through nuclear pores. So जो भी passage है substances का इन pores के through होता है. And nucleoporins are the proteins which assemble into a circular nuclear pore complex, जो बनाती हैं this circular complex जिसे बोलते हैं हम nuclear pore complex. इसका diameter होता है, it ranges from 10 nanometer to 100 nanometer, means uniform size का नहीं होता, it varies. Mammalian cells में typically present होते हैं लगभग 3000 nuclear pore complexes, varying based on cell type and life cycle of the stage. Life cycle stage means ये vary करता है cell type पे ऊपर और cell के life cycle पे. Next, this nuclear pore complex is cylindrical, multi-protein complex that surround each nuclear pore. और जो nuclear pore complex होता है इसका molecular weight होता है 60 to 125 megadalton, and it consists of 30 different nuclear proteins. बहुत तरह के proteins present होती हैं, that's why ये complex है. और इसके लिए एक term use की जाती है, that is nucleoporins और NUPs. इन्हें हम NUPs भी बोलते हैं इन proteins को, या फिर nucleoporins भी बोलते हैं. In eukaryotic cells, NPC is termed as gatekeeper of the nucleus, as we know that it controls the flow of substances, that's why it acts as a gatekeeper. The structure of nuclear pore complex comprises the following key elements. Firstly comes nucleoporine scaffold. So it constitutes the formation of nuclear pore complex, and these proteins entangled massively to permit selective exchange within the cell and it contains cytoplasmic ring जो कि बाहर की तरफ present होती है. This is cytoplasmic ring. This ring is known as cytoplasmic ring. Then it contains nucleoplasmic ring which lies towards the nucleus. This ring then it contains central spoke rings that we can see here. These are central spoke rings. Then this NPC appears as an octagonal due to scaffolding of few nucleoproteins. ये दिखता है हमें octagonal, means having 8 sides. The next comes central channel. So this is the central channel, this one. It is 36 to 38 nanometer wide. इसका जो dimensions है 36 to 38 nanometer wide, and is covered by 8-fold symmetrical spokes. और ये cover होता है इन spokes से. It is also known as transporter. इसे transporter कहा जाता है क्योंकि जो exchange of material है वो यहीं से होना है. Some nucleoporins like we have these repeats which are known as FG repeat. F means phenylalanine and G means glycine. तो ये जो amino acids हैं ये इनमें बहुत ज़्यादा होते हैं. So these proteins contain FG repeats and are responsible for the formation of channel, जिससे बनता है channel. जो FG nubs हैं they are characterized by the domains with extensive repeats of these two amino acids which are known as phenylalanine and glycine. These functions as selective barrier. ये इनका जो main role है, they act as a selective barrier and only allows both import and export of large biomolecules across bilayer nuclear envelope. तो selective exchange of material में important role play करते हैं. The next important structure in nuclear pore complex is cytoplasmic filaments. तो ये जो filaments हैं जो कि cytoplasm की तरफ present होते हैं, which rises from this ring which is known as cytoplasmic ring. These are known as cytoplasmic filaments. ये काम करते हैं as a sensor or receptor की तरह काम करते हैं. So 8 in number. These are 8 in number. 8 होते हैं number में, and cover less space towards cytoplasmic end. और इसका जो diameter that is 3.3 nanometers. They appear as a short and thick tough structures associated with this cytoplasmic ring. ये जो structures हैं ये associated होते हैं इस ring के साथ, that is cytoplasmic ring, and these are composed of nucleoporins. We have special nucleoporins named as NUP88, NUP214 and NUP358. So ये जो specific proteins हैं ये इसमें present होती हैं, और इनका जो main function है cytoplasmic filaments का, they act as sensors. ये act करते हैं sensor की तरह that specifically bind with signal proteins, tagging molecules that have to be imported into the nucleus. जो कि nucleus के अंदर import करने हैं उनके लिए ये receptors की तरह काम करते हैं. Next structure is nuclear basket. So this is nuclear basket, this entire structure. So this is a large basket shaped structure associated with nucleocytoplasmic side of the pore, and it's made up of ring of nucleoporins like we have NUP153 and NUP50, which bind filaments of TPR, that is tetra triacopeptide repeat structural motif. ये specific motifs present होते हैं इसमें protein to form a basket shape structure जो help करता है इस structure को बनाने में, basket shape structure को बनाने में. It enables tethering. Tethering means to bind of nucleoporins inward the nuclear ल्यूमन। तो ये जो प्रोटीन्स है ये टेथरिंग में इंपॉर्टेंट रोल प्ले करती हैं। टाइंग में मीन्स ये जो बास्केट लाइक स्ट्रक्चर बनता है इसको बनाने में हेल्प करते हैं। इट प्लेज अ सिग्निफिकेंट रोल इन एक्सपोर्टिंग बायोमोलिक्यूल्स। तो जो बायोमोलिक्यूल्स न्यूक्लियस के अंदर बनते हैं उनको बाहर ले जाने में ये इंपॉर्टेंट रोल प्ले करते हैं। The next structure is nuclear ring. So this nuclear ring contains four parts, that is the membrane ring, then the spoke ring, inner ring, and the outer ring or cytoplasmic ring. So NUPs classified into three categories. We have three different categories of these nucleoporins or NUPs. पहला है transmembrane nucleoproteins जो बनाती है लगभग 10% of this nucleopore complex. Then we have some structural nucleoporins जो बनाती है 50% of this part, and then we have this FG repeating NUPs जो बनाती हैं 40% of this nuclear pore complex. So this nuclear pore, these nucleoporins in nucleopore complex and their associated exportins and importins are also called as karyopharins. इन्हें हम karyopharins भी कहते हैं and are responsible for nucleocytoplasmic transport, और इनका जो main role है that is exchange of material between cytoplasm and nucleus. So proteins are involved in cell differentiation. इनका जो important role है ये cell differentiation में भी help करती हैं. Then molecules smaller than 40 kilodalton, जो इतने साइज के molecules हैं वो freely pass कर सकते हैं in nuclear pore complex से, while जो larger biomolecules है they are transported actively. उसके लिए फिर ATP use होता है for its transport. The next important part of nucleus is nucleoplasm. It is basically a matrix which is present inside the nuclear membrane. So this structure which lies inside the nuclear membrane is known as nucleoplasm. So this is a suspension fluid that holds the chromatin and nucleolus. दो चीज़ें इसमें present होती हैं. These fiber-like structures जिन्हें हम कहते हैं chromatin, and this dark stained structure that is known as nucleolus. So this sap-like structure is also known as nuclear sap or karyoplasm. It contains primarily water, dissolved ions, and complex mixture of molecules. It contains the important structures like DNA and RNA in the macromolecular state and in the form of their monomer nucleotides. तो इसका जो raw material है वो भी इसमें present होता है. It also contains both basic and acidic proteins, and these basic proteins plays very important role in DNA packaging. The most important basic proteins of the nucleus are nucleoprotamines and nucleohistones, which are very important for the assembly of DNA, or we can say packaging of DNA. So the nucleoprotamines are simple basic proteins of low molecular weight, that is 4,000 daltons, and arginine is the most abundant amino acids which are present in this proteins, that is nucleoprotamines. इनमें जो सबसे common amino acid पाया जाता है that is arginine, और इसका जो pH है that is 10 to 11 means these are basic proteins. इसमें ये functional होती है. Next we have these important proteins which are present in the nucleoplasm, that is nucleohistones, which are basic proteins and are composed of the basic amino acid arginine and lysine. Means ये जो proteins होते हैं these are rich in these two amino acid, that is arginine and lysine. इसमें ये ज़्यादा अमाउंट प्रेजेंट होती हैं. इनका जो मेन रोल है दैट इज इन डीएनए पैकेजिंग और डीएनए सुपर कॉइलिंग. सो मोस्ट ऑफ द न्यूक्लियर एंजाइम्स आर कंपोज्ड ऑफ नॉन हिस्टोन एसिडिक प्रोटीन्स. द मोस्ट इंपॉर्टेंट न्यूक्लियर एंजाइम्स आर डीएनए पॉलीमरेज, आरएनए पॉलीमरेज, देन वी हैव एनएडी सिंथेरेज, न्यूक्लियोसाइड ट्राइफॉस्फेटेज, एडेनोसिन डाई एमिनेज, देन एल्डोलेज, इनोलेज, 3-phosphoglyceraldehyde dehydrogenase and pyruvate kinase. So ये जो सभी के सभी एंजाइम्स हैं ये भी प्रेजेंट होते हैं न्यूक्लियोप्लाज्म. Then this nucleoplasm also contains certain cofactors and coenzymes such as we have ATP acetyl coenzyme A. Then it also contains lipid content and some inorganic compounds such as phosphorus, potassium, sodium, calcium, and magnesium. Its primary function is to act as suspension medium for organelles of the nucleus. So nucleus के अंदर जो organelle उसके लिए suspension medium की तरह काम करता है. It maintains the nuclear shape, that is very important function which is carried out by this matrix, and the transportation of ions, molecules, and other substances important to cell metabolism and function. So this is all about this nucleoplasm. The next important component of nucleus is nucleolus, which is a darkly stained structure present in the nucleus and is also known as the little nucleus or the brain of nucleus. This was firstly observed by Fontana in the skin cell of eel, and the term was coined by Bowman in 1840. It is also known as little nucleus. So we can see here to be a central portion, and that is occupied by the structure which is called nucleolus, and it is a dense rounded dark staining organelle inside the nucleus. It is without a limiting membrane and occupies about 25% of the volume of nucleus, and it is mainly involved in assembling the ribosomes, modification of transfer RNA, and sensing cellular stress. So these are the activities which are carried out by this cell only. That is assembly of ribosome, modification of tRNA, and sensing cellular stresses. So the cells of bacteria and yeast lack this organelle nucleolus. तो ये absent होता है in bacteria or yeast cell में, and the size is found to be related to the synthetic activity of the cell. So metabolically active cells में इसका size बड़ा होगा as compared to the cells which are in resting stage. So cells like we have sperm cells, blastomeres, muscle cells. In these cells, there is little or no synthetic activity, so they have smaller or no nuclei. While we have some cells like the oocytes, neurons, and secretory cells, which are metabolically active cells, which synthesizes the proteins or other substances, they have comparatively larger sized nuclei. तो ये जो cells हैं, oocyte है, neuron है, इनमें जो size होता है nucleolus का वो larger होता है as compared to the cells which have little or no synthetic activity. So the number of nuclei in the nucleus also depends upon the species and the number of chromosome. The number of nuclei in the cells may be 1, 2 or 4. तो ये number हो सकता है एक cell में nucleolus का, nuclei का. The position of the nucleolus is in the nucleus is eccentric, means away from center. So the nucleolus consists of 3 regions. 3 regions होते हैं. So the first region is the fibrillar region, which is the central portion of the nucleolus. So fibrillar center, and this is central region of much denser fibrous network. It is also known as pars fibrillosa or nucleonema. तो ये terms भी इसके लिए coin की आती हैं, and it's composed of large number of small fibrils of about 50 to 100 Armstrong in diameter. तो 50 से 100 Armstrong का diameter के fibril से ये बना होता है. These fibrils represents the long rRNA. Here represent करते हैं rRNA and precursor molecules in the early stages of processing. So, ये region बनाता है fibrillar region, जिसे बोलते हैं pars fibrillosa और nucleonema भी. Here we can see here the central portion, that is the fibrillar portion. It forms this fibrillar. So the next part in nucleolus is granular region. So it contains small darkly stained structure granules having a diameter of about 150 to 200 Armstrong, and these lie scattered in the amorphous matrix. They are also known as pars granulose. इस part को pars granulose भी कहा जाता है, और इसमें जो ratio होता है protein और RNA का, that is 2:1. These are the precursor of ribosomes. Then the third region is amorphous region. So it is an outermost region in which the granular and fibrillar regions are suspended. It is also called pars amorpha, and it is a homogeneous ground substance of the nucleolus, which is composed of fibrillarin protein that is associated with RNA processing. The most important part of a nucleolus is nucleolar chromatin. This is nucleolar DNA, which is 100 Armstrong thick chromatin fibers and contains many copies of DNA that directs synthesis of ribosomal RNA, जो कि rRNA बनाने में help करती है. इसमें जो proteins होती हैं which are known as nucleophosmin proteins, and these are responsible for transport of fluid between nucleus and cytoplasm. It also helps to carry out various cellular functions like protein formation, DNA replication, and cell cycles, which are carried out by nucleolar fluid. ये इन चीज़ों में important role play करता है. Then there is one important region that is called nucleolar organizer region. In chromosomes, normally there is one constriction which is known as the primary constriction. This is called primary constriction or centromere. In addition to this, sometimes there are some other constrictions which are known as secondary constriction, and these secondary constrictions are also known as NOR, that is nuclear organizer regions, and these are the sites of rRNA synthesis. So these sites act as the formation of nuclei during interphase, and these are known as NOR, or nuclear organizer region. Chromosomes which bear NOR, that is nuclear organizer region, are known as nucleolar chromosomes. And there is at least one nuclear chromosome in the haploid set of chromosomes, sometimes more than one. For example, in maize, chromosome number 6 and 9. In case of humans, chromosome number 13, 14, 15, 20 and 21 contains NORs. So approximately जो haploid set है उसमें एक NOR होना ही चाहिए, एक से ज़्यादा भी हो सकता है. The next comes function of nucleolus. So it is considered as the brain of nucleus and is mainly involved in production of subunits that forms ribosomes. So ribosomes का raw material यहाँ पे बनता है. It is involved in ribosomal RNA synthesis. RNA की जो site of synthesis है, that this is nucleolus, and the formation of ribosomes in eukaryotes. It is also function in the assembly of signal recognition particles and the cell stress response. So these are the various functions carried out by a nucleolus. This was all about for our today's discussion about nucleus. I hope you will get some idea from this presentation. If you have any questions, queries, you can ask in comment section. Thanks for watching. Have a nice time.